So the question is, why would this protein go away like this? And what could be the cause? So my advisor and his team of scientists, what they did was they used a set of protease inhibitors. Okay. And a protease inhibitor is just that. It inhibits a protease. So it clogs up a protease and stops it from chopping up an enzyme or a substrate. So when you have a protease inhibitor, it's like a fingerprint. You know which protease it's going to inhibit and clog up. So this one, the lactocystin, that's what that LC stands for, and this MG132, these all basically pointed to the 26S proteasome system. Okay. And that's what this set of experiments in these cell lines basically told them. So they saw it go away. They saw guanabens accelerate a loss of the protein in the rat. And now in the cell line, they saw it accelerate in the cell line. And with these protease inhibitors, this suggested that it was due to a system called the 26S proteasome. Okay. So I did not explain what a protease is and why this all matters and how this all fits together. So a protease, so an animal cell, our bodies are made up of billions and billions of these. And depending upon the tissue type, the cells have particular functions and particular properties, but the majority of our cells have a nucleus and a cytosol and a cell membrane and ribosomes and a Golgi apparatus and mitochondria. But also in that cytosol, there are peroxisomes and there are proteases. I don't see any proteases in this cartoon, but what a protease does is it basically cleaves a protein. And so our proteins, our cells have mechanisms for creating proteins, and then there are housekeeping mechanisms for degrading and cleaving those proteins once they've got to the end of their life cycle and once they have outlived their usefulness. So our cells have mechanisms for creating proteins and mechanisms for degrading proteins. Okay. And those two arms work hand in hand. And it's theorized that we know that when any of those mechanisms break down, when any of the natural mechanisms in the cells break down, that's when disease states start. But when it's been postulated that when certain pathways break down, and for example, when proteins become misfolded or unfolded, and when they build up in cells, that can lead to cancer, and that can lead to disease states. In any case, the thumbnail for this live stream and where my the last paper I showed you concluded was this. This is the 26S proteasome system. So this is and this is from about 15 years ago. So science has evolved, and there may be more known about this now. But this is the 26S proteasome right there in the center. That is it's a protease system. Those purple UBs, those are proteins called ubiquitin. And basically what you need to know is that in order for a protein to be targeted for cleavage and digestion by this proteasome here, that protein has to be tagged with these ubiquitin proteins. And those are tagged by ubiquitinating enzymes represented by E1, E2, and E3. So once they're tagged with those ubiquitins, they are recognized by the proteasome. The ubiquitinating enzymes take the ubiquitin off, and the protein is cleaved into little bits, and the amino acids are recycled so the cell can use them. And this is an ATP-dependent process on the front end and on the back end. So that last paper I showed you suggested that our enzyme nitric oxide synthase was degraded or is degraded in the cells by this proteasomal system.